Good morning, good afternoon, good evening, everyone. Thank you very much for joining us for this session. My name is Stoyan Stoichev, and I'm part of the ReSyn Biosciences team. Today we have two great presentations from Professor Sergey Vakhrushev, based at the Copenhagen Center for Glycomics at the University of Copenhagen, as well as Dr. Vincent Geoghegan at the Center of Immunology and Infection at the University of York. Before we get into the presentation, I just want to give you a very brief overview of who we are, what ReSyn Bio is all about, and some of the latest developments in our space, and then we'll jump in straight into the two presentations. So ReSyn Bio is all about improving the efficiency and throughput of mass spectrometry and bioseparation workflows using a high capacity and specificity magnetic microparticle technology. So we've developed a novel technology that allows us to bind throughout the surface of the microparticles rather than just on the surface. This then translates on several advantages and benefits, including increases in binding capacity, irrespective of which chemistry we use on a microparticle, allowing us to miniaturize reactions and improve the sensitivity of workflows. We also have very high functional group density, which allows us to do multi-point attachments of macromolecules, reducing their leaching as well as preserving their activity. Very early on in the product development cycle, we introduced magnetics into our products, and we believe that high performance magnetic separation is a very efficient way to transition from manual to automated workflows, as well as scale your workflows without the requirement for major rework of your methods. We currently have 15 products on the market that can be broadly separated into three areas: the bioseparation, immunoaffinity type of capture, the proteomics, which includes phosphopeptide enrichment products, enzymes for immobilized enzymes, as well as a range of products for protein and peptide capture, cleanup, and on-bit digestion, including our new hydroxide microparticles, which we hope to have on the website in the next couple of weeks. So have a look at that at resynbio.com. We also have solutions for covalent capture of targets, and we provide custom support to clients and collaborators, which require different chemistry put in the microparticles, or they might require further modification of the current products in order to better fit the application, which you can also do. In 2020, the major shift for us was the introduction or the expansion of our IMEC range, where we introduced the high-performance titanium IMEC and zirconium IMEC microparticles. And throughout the year, we started seeing the first data sets showing the use of these products for high-throughput automated studies where you've got large cohorts, large sample sets to process. We further optimized the efficiency of zirconium IMEC enrichment to make it as good as or even better than titanium IMEC, giving us additional selectivity. And we're starting to see the application of this chemistry for phosphopeptide enrichment also for lower input materials. Something a little bit outside of the phospho world, and a good recent examples of our ability to do custom development work is where we modified and stabilized the hydroxide microparticles in order to make them compatible with the very harsh conditions used for proteinase digestion. This in turn enabled the ability to do on-beat proteinase digestion, which improved the digestion efficiency of this enzyme in comparison to in-solution digestion. In 2021, we expect to see a further push in terms of the requirement to use lower and lower input material. And we're starting to see some data sets being presented and published in 2021. A good example is the one where we see the use of our workflows and products for total proteome and phosphoprotein analysis of subcellular fractionated samples. The new WISPR methodologies from Evocept are really pushing the ability to do low input phosphopeptide enrichment, and in combination with our high performance titanium IMEC microparticles, we're seeing now the ability to enrich for inputs for as low as 1 microgram. And these workflows are still automated, so you can retain the ability to do high throughput analysis, as well as the ability to have high reproducibility across your sample set. A little bit outside of the phospho world, outside of the proteomics world, we also have impact into biocatalysis applications. And this is because our platform is very well suited for the stabilization of macromolecules such as enzymes, where we can not only stabilize monomeric, but also multimeric enzymes, preserving the activity due to the multi-point attachment of the technology. In 2021, we hope to further expand our high-performance product range. We're specifically aiming to further improve some of the products in order to couple them better to LC-MS applications. So this means more narrowly distributed microparticles, improving selectivities by changing the backbone, as well as protecting the immobilized biomolecules, so one can do on-bit digestion without generating contaminating peptides that will interfere with downstream LC-MS. So if you are interested in these type of products, we do have prototypes in development. Come and talk to us and we can chat about how we can test these. So now I'd like to switch to the main part of the session and introduce our first speaker, Sergey Vakhrushev. And Sergey is a principal investigator and group leader at the Glyco-MS group at the Copenhagen Center for Glycomics. He completed his PhD in 2006 from the University of Munster. And his main research interest is fundamental studies in glycobiology and the development of mass spectrometry-based methods for glycomics and glycoproteomics using DDA and DIA-based workflows. His focus in the clinical development is in congenital disorders and cancers using targeted glycoproteomics as well as GlycoDIA methods. And today, Sergey is going to tell us a little bit about his development of GlycoDIA approaches for all glycoproteomics profiling. So Sergey, the floor is all yours. And just stop my share.
Yes, thank you, Stoyan. Thank you, the committee, for the opportunity to share some of our results. Just 1 second. Where is the screen? Yes. Do you see my screen, Stoyan? Is everything okay?
Yes, I can see your screen.
Yes, perfect.
Go ahead.
Yes, I go. So thank you again. Thank you for the introduction. Thank you for the opportunity to share some of our results. I'm heading the Glyco-MS group at the Copenhagen Center for Glycomics, which you see on the right corner on our slide. And today I'm going to talk how we can fuse two technologies together, which are gene editing and advanced mass spectrometry, to explore glycoproteomics field or glycoproteomics. Today I'm going to talk only about all glycoproteomics. The glycoconjugate landscape is depicted on the schematically on this slide, and our focus today is going to be about O-Gal-NAC type glycosylation. O-Gal-NAC type glycosylation is super heterogeneous, and key factor is that it's initiated by this differential panel of 20 GalNAc-T isoenzymes, which are differentially expressed in different organs. And the key feature of this type of glycosylation that there is no really consensus motif like in glycosylation, and as I said, it's very differential and dynamic. So this is our focus. So why O-Glycosylation? I just would like to give you a few examples why O-Glycosylation is important. One example is cancer or tumor-associated O-Glycosylation. In these two slides, you see how the O-Glycosylation landscape could be variable depending on the healthy or tumor conditions. So in case of the tumor conditions, the O-Glycosylation tends to reduce its complexity, and the nature express some epitopes which are usually not present in the healthy control. Those on the bottom, these simple epitopes like TN, SIL-TN, and T epitope. So another example of the importance of glycosylation is the fine-tuning of site-specific glycosylation. As you can see on a couple of these examples, the location of the glycosylation or O-Glycosylated type glycosylation enhance or influence on a certain properties of the protein, like ligand binding, ectodomain shadings, receptor dimerization, and PC processing. So that's why it's so important to understand and to know the location of this type of sugars. How to do it? As I said, it's extremely complex, and it's not easy to study. And we decided to introduce the concept which we called a SimpleCell technology, where we used gene editing technology, at that time using a finger. This time we're using a CRISPR knockout strategy. If you look on this picture, how the biosynthetic pathway is formed, in order to elongate O-Glycosylation machinery, you need to have such a chaperone called cosmic, which activate the activity of the glycoenzymes, which is C1-GLT1 enzyme, which place galactose on the initial GalNAc, and then other enzymes can build the structure further. If you knock out this enzyme, this chaperone, then enzymes stop working, and the whole biosynthetic pathway would be disturbed, and instead of a full variety of structures, you would express only simple epitope like TN. So this is the core of our technology. We call it SimpleCell. So the analysis is done by using mass spectrometry. So SimpleCell is to generate a sample. Then we introduce a lectin weak affinity chromatography to enrich corresponding glycopeptides. In this picture, we have VVA. VVA is the lectin, which is specific for the TN epitope. Then we apply mass spectrometry with HCD or ETD fragmentation, and we run a standard data-dependent acquisition to identify cases in a site-specific manner. So we some years ago we applied this technology on a different. We tested on the different simple cells. At that time, we used 12 different simple cell covering organs, different organs, as you can see in the picture. And we managed to obtain a very solid and deep data, which was at that time a kind of a breakthrough in the field. And we managed to maximize the knowledge about O-Glycosylation type of glycosylation. Later, we introduced a few more lectins into the panel, such as PNA or Jacqueline. Those two lectins are highly specific to the epitope, which is depicted on this picture. It's specific for terminal galactose, and this actually enabled us to enrich a population of glycopeptides from the non-simple cell, but from the real samples. So if you think that a lot of GalNAc expressed as a sialyl-T form or disialyl-T form, if you do a simple cyridase treatment or action by the neuraminidase and remove a terminal sialic acid, then you enable this epitope for the lectin recognition. And if you apply this sample to the PNA or Jacqueline enrichment, you can pull out a quite substantial amount of glycopeptides. Applying this technology, I just give you a summary of our current data set, what we managed to obtain. So currently in our database, we have about 15,000 glycosides unambiguously assigned, which correspond to approximately 5,000 proteins called to be glycoproteins or GalNAc glycoproteins. This is our discovery platform. And as you can see, immediately we faced the question that what do we call to be glycoprotein? So an important factor now to understand what is the stoichiometry level or what is the occupancy level of the glycosylation. So we would like to understand the amount of glycosylation placed on a certain location or position. At the same time, we face the problem that in many applications, the amount of material is extremely limited, and you also need to do something with that. We started a new project where we implement data-independent acquisition into our pipeline, which help us to address those listed questions like sensitivity, stoichiometry, and perhaps even direct glycoproteomics. So now I'm going to talk a little bit more about that. So on the current slide, I would like to show you the workflow for the GlycoDIA method we called. As in standard DIA approach, first what you need to do, you need to build a set or collections of reference spectra. Reference spectra which you are going to use in your future DIA analysis. So we applied samples, we did a very deep laborious protocol, a lot of LC runs, and we built a library. So in this case, I just want to show you that a summary of library which consists of HEPG2, HEC, and some ovarian cancer cell lines, and also some wild type, and also we used human serum and plasma, so which is a part of our library. And when you have this library, then you can apply this data set to your analysis of your data-independent data. Just briefly speaking, data-independent acquisition, in contrast of data-dependent acquisition, is a method where instead of selecting individual precursors in a very narrow window, instead you select a certain range in your ion current in your scan, so which gives you a capacity to screen more potential precursors, but at the same time it complicates your data analysis because instead of having one MSMS per precursor, you have kind of a chimeric MSMS, or a mixture of fragments which correspond to various precursor ions. But this is exactly how DIA works, and the library help us to perform the identification. This is the workflow summary, and then we applied this summary into our pipeline. So the key feature I would like here to mention that there is an interesting phenomenon in glycosylation, that due to the very high nature of, let's say, the high rate of instability of such PTM on a peptide backbone, what you typically observe doing HCD type of fragmentation is that your sugar is extremely labile and it cleaves immediately off, and then your fragments are mostly representing a naked fragment. So that means fragments with the sugar neutral loss. So if you compare, for example, two spectra, T spectrum, T glycopeptides on the bottom, and TN spectrum on the T spectrum on the top and TN spectrum on the bottom, that you can see that they're highly similar. So all peptide fragments are nearly the same, except you get a little difference between oxonium fragment ions. So we made a test, and we compared a set of 6 top intensive fragment ions from 2 different glycopeptides, and you can see on this corner that they're highly similar. So this actually inspired us to come to the hypothesis that fragmentation pattern of distinct peptides with different glycans are identical. And we made a couple of tests where we found a very high correlation across DIA spectra from either replicate or between T and TN, even naked, sharing the same sequences, sharing the same peptide sequences. So taking this as a fact, we come up with a hypothesis that if we start with experimental library, which is here on this slide, showing, for example, TN or TN, you can use the same spectra for different glycoepitope, which is difficult to build experimentally, such as sialylated forms. It's not that easy to enrich for sialylated forms. That's why it's extremely difficult to build those type of reference spectra experimentally. So, but if you do this in silico reconstruction, just simply modifying the precursor ion, that you can use the same set of fragment ions for other glycopeptides. At the same time, we can do the opposite. We can do subtraction, so we can remove sugars from the reference spectrum and then try other more reduced candidates. For example, since we're interested to understand the stoichiometry, we would like to see the ratio between glyco and non-glyco forms. So we take glyco spectrum as a reference, and then we remove a sugar, and we create a virtual naked spectrum, which could be used for identification of naked peptides, and we can apply that in our pipeline. So this is the technology, and now I'm going to show you a couple of applications. So at first we try to apply this to the direct analysis of human serum. So we took 10 microliters of human serum, and we made a top 2 depletion, so removal of top 2 most abundant protein. Then we did a triptych digest. We treat sample with sialidase and no sialidase, and injected 2% into the mass spectrometry directly. So no enrichment, just directly LCMS. So, and here, this is the list. So we managed to identify with our approach approximately 159 glycoproteins, which are all manually validated. So we went through every single case, and on the corner, you can see the overview of the identified glycopeptide. So you can see that with this approach, we could identify TN glycopeptides, T and sialo-TN, sialo-T and disialo T. So 5 different glycopeptides using just one single library. So quasi this corresponds to the extremely low amount of plasma serum, like 2 microliters. So in this example, I would like to show you our test. We try to evaluate what could be the site occupancy reported by this method. And you can see that we decided to cluster occupancy into the 3 categories: high occupancy, medium occupancy, and low site occupancy. And here you see 3 examples where we could quantify glycopeptides and rank them according to this occupancy. Next, what we are trying now to develop in this approach. So as I said, we're suffering a lot with the limitation of the amount available for the analysis. And we decided to introduce magnetic beads into our enrichment pipeline. So we call it a kind of Glycoproteomics high throughput pipeline, where we integrate GlycoDIA, we integrate a mini LVAC approach, and potentially we want to move everything on the chip, so which would be a lab on chip kind of glycoproteomic solution. So how this is working? So you can see LVAC approach, canonical LVAC approach, based on the lectin column, which is usually like 2 meter long column and requires some HPLC system. Instead of that, we shifted conjugated magnetic beads into the lectin and used that for enrichment of glycopeptides. In this case, we conjugated Jacqueline lectin with a magnetic bead and applied this for the analysis of 2 microliters of human plasma previously treated with neuraminidase. And that's what we could achieve using this approach in a quick manner. With this approach, we managed to identify, confidently quantify approximately 1,000 unique glycopeptides. So, and which correspond to approximately 179 glycoproteins. So this pipeline give us a very high hope or potential to extend our technology further and perhaps probe to do glycoproteomics on a single cell level, or let's say on the extremely low amount of cells, and also use this into the clinical glycoproteomics approaches. So I'm done with the lecture. So I would like to conclude that the technology is ready for the next generation glycoproteomics, where you enable to do analysis of sugars simultaneously, site, structure, and occupancy, GlycoDIA could be explored for other type of glycosylation by in silico extension. It has a perspective for clinical glycoproteomics. At the same time, we faced that there's high demand or high necessity to improve and work further for the improvement of false discovery rate identifications in DIA, and also ETD for DIA. And I would like to acknowledge our center, Copenhagen Center for Glycomics, and head of our center, Henrik Clausen, my former colleagues, PhD student Zilu, Yang Mao, Katarina Stentov, and Tomislav Zaval. And I would like also to thank ReSyn Biosystems and Stoyan and Justin for their support with magnetic beads. And thank you for your attention.
Thank you. Thank you very much, Sergey. It was a fantastic presentation. Just to the participants, please submit your questions. We have a couple of questions. We're going to get to those after the second presentation from Vincent, but keep submitting the questions and we will try and answer them at the end of Vincent's presentation. So I would like to then introduce our second speaker for today, and this is just share my screen. Dr. Vincent Geoghegan from the Center of Immunology and Infection, University of York. Vincent received his PhD from the University of Oxford in the UK, and he was focusing on investigating T lymphocyte activation through the use of novel enrichment methods for the in-depth analysis of arginine methylation. He later moved to Lancaster University and investigated host responses of Wolbachia in an effort to understand how this endosymbiotic bacterium blocks viral replication in mosquitoes. He is currently at the University of York and is applying his expertise to understand the role of kinases in the Leishmania cell cycle. He is establishing proximity or biotilation approaches to study the system in order to capture the interactomes that previously have been beyond our reach. So Vincent, you can go ahead. The floor is yours.
Okay, thank you, Stoyan. Okay, so thanks. I'm going to talk today about the work I've been doing looking at kinetochore kinases in Leishmania in the Mottram lab at the University of York. So the Mottram lab, we focus on this parasite called Leishmania, that's here, and it's a unicellular flagellated eukaryotic parasite that's transmitted by these blood-sucking sandflies, and it causes a range of human and animal diseases called the Leishmaniasis. And here you can see some patients that are affected by cutaneous and mucocutaneous Leishmaniasis. So looking at the caseload of Leishmaniasis across the world, so this is a map of the prevalence of visceral Leishmaniasis, which can be lethal, and we see that the sort of burden of disease falls really on the less developed countries sort of around the equator, and this causes about between 20 and 30,000 deaths per year. And we can expect that as the climate changes and the range of this sandfly vector expands, then more and more people are going to be affected by this parasite. So really, there's quite an urgent need to develop a better sort of molecular understanding of this this parasite that causes this neglected tropical disease, Leishmaniasis. So to help with that, I've been looking at the kinetochore, which is a large protein complex in all eukaryotic cells, and it's a critical protein complex because it binds to this specialized region of chromosomes called the centromere, and its role is to connect, to link up chromosomes to the spindle microtubules during mitosis, so that the cell can equally portion its chromosomes to opposite poles, so you get proper chromosome segregation. And it's a very large protein complex. So on the right here, there's a model and an EM image actually of the kinetochore complex in yeast. So we have the inner kinetochore, and that's the part that binds the centromere region of chromosomes, and then the outer kinetochore, which binds the microtubules of the mitotic spindle. So what about the kinetochore in Leishmania then? Well, in other eukaryotes, so here, for example, I've got the human and yeast kinetochores, and you can see there's quite a lot of similarity and there's homology between the components of kinetochores in many eukaryotes. But when the Leishmania genome was first sequenced, homologs of these kinetochore proteins couldn't be found. But we knew that Leishmania and related parasites had to have a kinetochore so they could properly segregate their chromosomes during mitosis. So that remained an open question until 2014, when Bungo Akiyoshi in the Gull lab managed to identify a set of what he called kinetoplastid kinetochore proteins, so the KKTs, in this parasite Trypanosome brucei, which is closely related to Leishmania. So he identified this set of proteins, so that was a really great starting point. And amongst this set of proteins, there are 4 kinases, so 4 kinetochore kinases. So I took the homologs, I got the homologs of these kinetochore kinases in Leishmania. So 2 of them, KKT2 and KKT3, are actually constitutively located at the centromere on the chromosomes. And the other 2, KKT10 and KKT19, these highly related kinases, which are essential for the progress from metaphase to anaphase of the cell cycle. So I first tried to do just pull downs to try and find the interaction partners for these kinetochore kinases in Leishmania, but I had limited success. I think that was because the kinetochore in Leishmania is very difficult to solubilize. So I decided to set up proximity biotinylation, BioID, in Leishmania, and that's where you genetically tag your protein of interest, your bait, with a promiscuous biotin ligase. You then add biotin to set off the biotinylation of proximal proteins in vivo. So then proximal proteins are covalently tagged with biotin, and you can enrich them with streptavidin and identify them by mass spectrometry. I then discovered when I was setting up a BioID that actually I could incorporate a cross-linking step to form cross-linking BioID. So in this workflow, I first perform the in vivo proximity biotinylation step and to tag nearby proteins, and then I perform an in vivo limiting cross-linking step with this membrane permeable crosslinker DSP, and then I can harshly solubilize everything to really get out all members of the complex and pull them down with streptavidin and identify them by mass spectrometry. So comparing the 2, so on the left, so this is KKT3, that kinetochore kinase, which has been tagged with this biotin ligase. So here, just normal BioID, I can identify some members of the kinetochore complex, but the extra cross-linking step in XL-BioID has really substantially improved the coverage of this large protein complex. So I took the used the XL-BioID workflow and set up this large experiment where I have 3 parasite lines, 3 Leishmania lines expressing biotin ligase tagged kinetochore kinases, KKT2, KKT3, KKT10. And very importantly, for proximity biotinylation experiments, I have this control line which expresses a protein tagged with this biotin ligase, which goes to the nucleus. So same compartment as these proteins, but it's not part of the kinetochore. So this serves as a reference, so we compare enrichment of proteins in these lines versus our control line, and that's very important. So I did the XL-BioID experiment on these Leishmania lines, 3 replicates each, ripolysis, enriched proximal proteins with magricin streptavidin on bead digest, and then performed label-free quantification and scoring of proximal proteins with St. Q. So this produced an interactome of these 3 kinetochore kinases shown in yellow. And in darker blue, we see the kinetochore proteins that we're expecting based on homology to trypanosomes. I did some microscopy screening, looking at these unknown proteins, and managed to identify 2 new components of the kinetochore, actually. So one of them is shown here, the KKTA. So this is a microscopy localization of this new kinetochore protein across the cell cycle, comparing it to the known kinetochore protein KKT2. And you can see these distinct punctae in the nucleus that appear at S phase as characteristic of kinetochore proteins. So that was really just a static picture of the kinetochore in Leishmania. I wanted to go a bit further and look at how the kinetochore might be regulated. And we know in other eukaryotes that phosphorylation is a very key regulatory mechanism in kinetochore assembly. And so I wanted to try and use this proximity material and enrich for phosphosites. So this is what I did. So I did my XL-BioID workflow, enriched for proximal proteins, again with streptavidin. Then I took this peptide sample and split it. So 40% goes for my total proximal sample, and 60% I use to enrich for phosphopeptides with magricin high performance titanium iMac, which is specifically designed to perform well on low amounts of material like I've got here. And then I do label-free quantitation as before. So I set up this experiment using that workflow. And here, what I'm trying to do is actually look at the phosphorylation state of the kinetochore through the cell cycle. So I have synchronized my parasites with hydroxyurea, and I've got my control line, and I've got my kinetochore kinase. So here I've actually switched to using the mini turbo ID enzyme, and that allows faster labeling kinetics. So then I can take 3 points during the cell cycle and do 30-minute biotinylation. I do 5 replicates each, and I can have a look at, what is the state of the kinetochore at each of these points in the cell cycle. On top of that, the lab has had a very fruitful collaboration with Novartis, developing this inhibitor of these kinetochore kinases KKT10, KKT19. So this inhibitor AB1, which targets KKT10, KKT19, and it causes parasites to accumulate later on in mitosis at G2M. So we know one of these, the one target for this kinase is KKT2, as part of the kinetochore, but there might be others. So I was interested in using this inhibitor to try and discover phosphorylation signaling pathways at the kinetochore in Leishmania. So I added that as an arm to my experiment. So looking at, so this is looking at proximal phosphopeptides to the KKT3 kinase. And so you can see early on in the cell cycle, there's not really that much happening in terms of phosphorylation at the kinetochore. We can see a few phosphosites in the bait itself, so in KKT3 itself. Then as we move into S phase, we see a quite dramatic increase in kinetochore phosphorylation sites coming up. And again, in G2M, we see a large number of phosphorylation sites coming up. including some in the bait itself, but also we can see some KKT2 phosphorylation sites, which I mentioned as being a target for KKT10, KKT19. So looking at a heat map of those phosphorylation sites, we see that the two cell lines basically cluster quite nicely together. So this region top right, that's the control cell line, the BDF5 cell lines, they cluster together. And then we have a number of phosphosites that are proximal to KKT3. And interestingly, we have here in the AB1 treated sample, so I'm inhibiting that kinase, that mitotic kinase, we can see this cluster of sites proximal to KKT3 that are reduced. So having a look at that, so plotting those separately, at 4 hours after AB1 treatment, so inhibiting the kinase in S phase, we can see that there's not that much happening actually. So there's only one proximal, or one phosphosite that appears to be reduced by AB1 treatment. But then when we apply the criteria of just looking for proximal phosphosites, this one is no longer important because we screened it out because it actually isn't proximal to KKT3. And so what I plotted on the right here is the phosphorylation sites that are reduced upon KKT10, KKT19 inhibition, and I further filtered these phosphosites based on whether they're proximal to KKT3 or not. And so we can see, for example, there are a couple of phosphosites, including this one on KKT2, which we know from previous studies to be a target of KKT19. So to conclude, the cross-linking BioID method offers a way to characterize quite biochemically sort of challenging complexes. And I was able to use that to identify 2 new components of the kinetochore in Leishmania, and they're conserved in other related kinetoplastids. And further, I was able to demonstrate that I can use cross-linking BioID to actually probe the proximal phosphorylation sites to the bait protein of interest. And by combining that with treatment with inhibitor, I can actually build a sort of a spatially focused picture of what this inhibitor is doing at my protein complex of interest. And so I was able to show that this kinase inhibitor AB1 decrease the phosphorylation of the kinase target itself, so KKT19, but also a few other interesting kinetochore targets, including KKT2 and KKT4. So just to wrap up, thanks to the Mottram group, so Jeremy Mottram and other people in the group who are working on protein kinases, who have provided a lot of direct help to this project, and the people in the proteomics facility at the University of York, Adam Dow, Tony Larson and Chris. Thank you to funding agency, the Wellcome Trust. And I will stop there and take questions. Thank you.
Fantastic. Thank you very much, Vincent. That was a really great presentation. We do have some questions. I think probably a couple of minutes to tackle those. We had a couple of questions coming through on Sergey's talk, and I think Sergey has already addressed that. So maybe we'll go back to them. I just want to jump to one that's specifically for your talk, Vincent. There was a question from Sabayachi Babu asking, are you worried about losing identifications due to cross-linked peptides?
Yeah, so that's a good question. So when I was setting up the cross-linking, I did quite a lot of testing and optimization to reduce both the concentration of cross-linker that I was using and the time that I was cross-linking for, to basically get it down to really the minimal level of cross-linking that I needed, because I was aware of, actually at the time I was really worried about just capturing loads of non-specific interaction just by aggregating everything, and because because I've got the biotinylation and the cross-linking, I don't need to cross-link hugely. So I did do searches in my data set looking for cross-linked peptides, and I see very few, I think. But actually, I think that's one thing that I'd really like to do in the future, is to use because I use a cleavable cross-linker here, but to use a non-cleavable cross-linker and to try and actually enrich for cross-linked peptides to try and define and identify protein-protein interaction interfaces. I think that would be a really nice direction. Yeah.
Okay. And a follow-on question: does both biotin and cross-linking label okay? Will this cause some problem?
So what was that? The biotin and
does both biotin and cross-linking label on lysine? Will this cause some problem?
Yeah. Yeah, it's something I've been thinking about. So I biotinylate first, so the cross-linking won't interfere with that. But you can imagine that, yeah, some cross-linking sites will already be occupied by biotin. So yeah, you wouldn't necessarily capture those. But somehow the cross-linking is giving me quite a boost in the coverage of this complex. It could be, yeah, the cross-linker has access to lysines that, you know, the reactive biotin in vivo does not, and that's why I get better coverage. Yeah.
It might be that the proteins are folded, so you don't have the access to the biotin. So once you unfold it, then you can cross-link those sites potentially.
Yeah, potentially, but yeah, I mean, yeah, that could be. I mean, I do the cross-linking in vivo, but it could be that as I add the cross-linker, obviously the cell obviously starts dying, and then maybe the complex opens up a bit or it changes a bit, and then it can access sites that, yeah, the biotinylation could not. Yeah, that's true.
Okay, and then for Sergey, Sergey, you addressed most of the question. There was one about the use of DIA instead of DDA when you're building your libraries. So I was just wondering from my side, what about additional fractionation techniques, maybe not reverse phase, but like ion exchange or even hydrophilic affinity to basically go a little bit deeper? Is that an option?
Yeah, definitely, it's an option. So the more fractionation you do, the better it is. Especially, yeah, it helps a lot for this so-called direct analysis with no enrichment. Yeah, we tried high pH. So we're also trying to now to do also helic on fly. So definitely yes, like you reduce the complexity and you can boost. Yeah.
Okay, interesting. So I think all the other questions have been answered. We are running out of time, so I'm gonna call it for this session. I want to thank very much to Sergey and Vincent for taking the time and for preparing these presentations. And thank to all of you for attending the session. We'll see you with the rest of the conference.